Today we are joined again by Noah Marcus, and he and Luna and Tim are here to talk to us about microbes and living soil. So I'm going to hand it over to Noah.
Thanks, Liz. Thanks everyone for having us back. We appreciate it. We'd like to welcome back proprietor and head cultivator from Love Light Medicinals, and general soil wizard, Tim Hanrahan. Also, side note, Tim's going out on the speaking circuit and will be next featured in February at the Organic Cultivators conference as a panelist. Definitely think you guys should seek him out there as well. And then we'd also like to welcome my friend Luna Whitcomb. Luna is an all-organic living soil probiotic natural farmer and grower. She's a writer for Skunk Magazine and administrator for the Cannabis Education Group, Organic Cultivators, as well as for Skunk Magazine. She specializes in soil biology, homemade plant inputs, ferments, compost, extracts, enzymes, and organic acids. Luna's methodologies focus on plant health and vigor. Luna, as long as we're talking about you, can you take this really big concept, this huge word, microbe, that everyone likes throwing around now, and kind of put it into a very simple definition for us?
Yeah, sure. First of all, thank you so much for having me. I'm thankful for the opportunity. Yeah, so microbes. Yeah, let's get into it. So yeah, microbes is a super diverse word. It encompasses a lot of different concepts and a lot of different microorganisms. So microbe is a microscopic organism. There's five main categories. There's bacteria, there's fungi, there's protozoa, archaea, and viruses. And their functions, by definition, are to convert one compound into another compound. And they all convert different compounds, different organic acids or sugar sources into other organic acids and usable compounds to the plant, different phytocompounds, as well as phytohormones. Um, so that's the large definition kind of brought into a small explanation, but each one of these different categories turn one compound into another specific compound. And the range between each one of the categories is massive of what kind of life forms exist within each one, and the effect that they have on this huge range of organic acids and sugars. They create a huge spectrum of compounds in the soil and for the plants.
Okay. So to take a step back and even understand why we want them there, I think maybe we should look at how root absorption works and really what our end goal is, so we can kind of work backwards from there. Tim, can you walk us through like a real, kind of high level, not high level, like kind of brief explanation? Because I want to get really into kind of how we use microbes, but to understand it, what the root system's actually doing.
Yeah, definitely. So, I mean, common theory of, chemical agriculture is plants uptake ions. Plants can uptake ions, but it's not the preferred form of nutrition. They'd rather uptake plant metabolites or really super complex forms of microbes, and even or microbial metabolites. But we're even understanding through rhizophagy cycle and the fact that all plants have endophytes, that plants actually uptake microbes through the root system, and they will uptake certain microbes. They will excrete a superoxide, will dissolve their cell walls, they'll squish the nutrients out, they'll transport what's left of those microbes to a new place, send it out a root hair, and then start feeding them there and create start the whole system over again. So really the way that we've been looking at agriculture since, for at least 120 years or so is completely wrong. We really want plants to uptake microbial metabolites. They don't want raw forms of, plants can uptake raw forms of N, nitrates and ammonia and urea and other forms, but they don't really want it that way. They really kind of want it in an amino acid form or a peptide or a more complex compound that can actually cause the plant to increase its energy instead of decrease its energy. A lot of studies are showing that if you feed a plant with nitrate, it takes 10 parts of sugar, 10 parts of energy in order to convert that nitrate into an amino acid. So that's a huge energy drain on the plant. Plus, the plant needs 50% more water in order to suck that nitrate up into its system versus, uptaking amino acids in a form that provides. So it is really like, really everybody's got to throw out their ideology of how we've been feeding plants for a long time, and there's a whole lot of new research out there that shows it's all microbial driven.
There's many ways or many claimed ways, I should say, about going about, creating this microbial life in your garden. Luna, I know for some time you were using different bottled microbial products like Microbe Life, and then we had some discussion recently that you kind of moved away from that. Can you talk a little bit about why you were using it, what you hope to achieve, and now why ultimately you don't, and what you use in place of it?
Sure. So there are specific roles that specific microbes have. And when in the presence of specific organic acids, and I know that's very broad and undefined, but I can get into a literal example. So there was a specific microorganism called Rhodopseudomonas palestris, and this is an endophyte. An endophyte is a microbe, in this particular instance, a bacteria, that is taken into the cellular, like the cell structure of the plant, and creates an endosymbiosis. It creates a symbiotic relationship with the functions of the cell. This particular one is a photosynthetic endophyte. It's a photosynthetic organism, microbe that is integrated into the cellular structure of the plant. And what it does is it photosynthesizes and it creates another form of photosynthetic energy for the plant. So it increases the energy inside of the plant and helps with chlorophyll density and things like that. So an example, like in our bodies, we have our mitochondria, our powerhouse of the cell, right? As we call it. This is an example of an endophyte that has been evolutionarily integrated into our cellular structure. What I'm referring to is also an endophyte that has been integrated into the evolutionary cellular structure of a plant. And by introducing it, that specific endophyte into our root zone, I'm encouraging photosynthesis and the amount of energy created by photosynthesis in the plant. There's in the Microbe Life product that you had mentioned called Photosynthesis Plus, this endophyte, Rhodopseudomonas palestris. There's a number of other ones that are photosynthetic endophytes also are present in this product. I had this discussion with some of the people in the organic cultivators back channel. We have like an administrative channel that we discuss concepts and stuff. And Tim had mentioned that this endophyte, this bacteria, is actually really common inside of earthworm castings. I was under the impression that it was a very specialty microbe that needed to be cultivated, needed to be isolated, identified and cultivated as a germ and then plated on an agar to then be multiplied and applied to your plant systems. It turns out that's not true. It turns out that a whole bunch of different photosynthetic endophytes are present in earthworm castings, which I'm a huge fan of earthworm castings and their applications, their mesophilic processes. So my soil base is the organic matter in my soil is mostly earthworm castings. It's like 30% earthworm castings. And so I found that the introduction of the product was redundant and therefore it was wasteful. So I stopped using it.
Yeah, a lot of these microbes, a lot of these microbes are really endemic. They're really prolific in a lot of different places, and it's about providing the right home and the food source for them, and then they thrive.
Tim, is there ever really a reason for a bottle solution, or let's say you have non-living soil, right? How do you introduce these microbes in a bottle or non-bottle solution? What is the solution?
So I mean, in an outdoor situation, you just got to provide a home for them. It's shown that in most fields, a lot of these microbes just kind of appear. A lot of them are are endemic or they're really tough and they've learned how to make cysts and spores and survive really harsh conditions. And it's shown that if you provide multiple different species of plants for their root exudates, because really what microbes want, they want photosynthetic compounds, they want root exudates. That's what most of the microbes that will feed, will have plant benefit, feed off of root exudates. So having the most amount of diversity of plants and root exudates will now cause a huge, influx of food for microbes, and all of a sudden everybody will start waking up.
Hey,
You can
Tim,
use yeah.
Can I just jump in? Is it okay if we have people jump in when they have questions, or would you prefer that
Oh
they
yeah,
wait
that's
until
fine.
the end with
Yeah,
them?
just fine.
Okay.
I'm trying
I
to pay
think
attention
Lucas
to every
has
screen.
one
But
right now.
yeah,
No,
yeah.
that's okay. I think Lucas has one right now.
Hey, what's up?
Hey, what's up?
Howdy.
Hi, Luna. I'm sad you only were one day at the Ganchia program.
Hey,
I think,
Lucas, it's good to see you.
yeah, you too. I have so many questions, and I wanted to wait till the end, but then you already started with soil and stuff and worm casting. Do you have any recommendations already for recipes and stuff? Like, is this a thing? Is there some stuff you can recommend? I saw about Kleckerman's cook or something. Is this a thing?
Lucas, I can send you the recipe that Tim and Luna worked on together that we discussed previously. I'd be happy to shoot you that way.
Super,
But just want to
awesome.
DM
Yeah,
your
really
email
nice.
address.
Okay, awesome. That's it. I thought just some recommendations. So she was just talking about the soil. But back to the microbes. Sorry.
No, totally. I would like to get into, I feel like we might get into that a little bit because minerals play a large role in this. And part of the reason why I developed the soil mix that, Luna's been using the way it has is based on calcium. Calcium's been shown to, it seems in studies, calcium seems to be one of the nutrients that will show a greater diversity of microbial species in the soil. So it has a lot of benefits. But as far as inoculating the soil, if you're in a container mix, like using a Coots mix or something else, you always want to use the best form of worm castings or compost you can find. A couple different diverse sources are great. Ideally, you want to ask them for if they have any mineral analysis tests, because I know some compost can be incredibly high in sodium, which causes all sorts of other issues. So it's good to know what your inputs are going in.
Mm-hmm.
But the best compost or worm castings you can find, go out to take some forest, some leaf duff or some forest soils. There could be some issues with bringing in potential pathogen, pest insects that way. But typically, if you go into an environment where there's not a lot of green living plants, there's a lot of trees, you go into a forest, and you just take up some leaf duff and some of the healthy humus just underneath the top layer, you're more likely to, I feel like you're more likely to bring in a lot of beneficial microarthropods and some predators. And I've never really found much issue with bringing in a pest that way. If you're going out and collecting off of live plants and in a plant where, in a field where there's lots of living plants and stuff, and you start bringing some of that material inside, that could potentially have, pest and pathogen issues.
How would that compare, Tim? How would that and Luna also jump in on this, how would that compare to IMO?
That's the hotly debated topic.
Luna, you know I was going there.
Yeah.
So Luna and I have talked about this extensively, and I've definitely seen some collections that Chris Trump has done that is definitely, a fruiting fungi of some sort. I've definitely seen some random collections where you're collecting things. Luna, you want to take this? You've dug a little bit more into the microbes and bucor and all that fun stuff.
So those that, just to hit the break for a second, those that don't know, IMO is the idea of collecting indigenous microbiotic organisms through rice in your natural environment wherever you're located. Right, Tim? That make that English right.
Pretty much, yep.
Luna, what do you think?
Yeah. So the abbreviation IMO is Indigenous Microorganisms, and it is a KNF, a Korean Natural Farming input, where the intention is to collect indigenous microorganisms from your surrounding environment. they hope to accomplish this by boiling rice. They kind of flash boil it for a little bit, I think like 15 minutes, 10, 15 minutes, to not fully cook it, but to kind of like just create a media for microorganisms to land on and multiply from your native environment, so that you can then multiply them in your soil or in some kind of aerobic fermentation, or even anaerobic in some cases, and then introduce it to your soil. So there's a few things to understand about how microbes behave when I think we discuss this. I think that this concept is potentially collecting indigenous microorganisms. I think that the conceptual understanding is a little bit off. So first of all, microbes blow through the air. They're in the ground, they move all over the place. They've covered the entire world. Most microbes have covered the entire world. And the reason they're able to thrive or survive or be in particular areas is based on the composition of the soil that they live in, the presence of specific sugars, polysaccharides, and polyphenols that surround them, which gives them a space to live, as well as the pH and temperature of that area. So all of these conditions affect what microbes can survive where. And they've all pretty much moved all around the earth. So when you're putting out a collection of rice, these microbes that are in your environment, surrounding your soil, they move, they'll move, they'll get to your soil. If you're using native inputs that are in your environment, those microbes are already on the inputs, and they're already in your compost, and whatever is going to survive is going to survive. When you collect them, or supposedly collect them, they're competing with whatever biology is currently on the rice. So what that means is, when they're prepping the soil and flash boiling it, people understand boiling, or treating it with this high temperature as like pasteurization, right? Pasteurization is not sterilization. It is preparing it to be safely stored and consumed, but it is by no means sterile. There are many microbes that can survive these really high temperature conditions, like lower fungi, like Mucor, Aspergillus, Trichoderma. But also, when you introduce these really high temperatures to a lot of bacteria and protozoa, they create what are called cysts. And in this form, they're considered spores. It's where they protect themselves in this little casing. And they can withstand these really high heats. So when they get back into the environment that they can thrive in, these cysts, they shed, and the bacteria, these spores now become active and start to compete for food source. In this particular situation, they're competing for starch, which is a polysaccharide. It is a sugar source. Not all biology wants the same kind of food. So this particular sugar source will only feed a certain spectrum of biology. So you're not creating a mirror image of nature or what you have in your environment. You're collecting a portion of it, or hoping to. Whatever blows in from the surrounding area is now competing with whatever microbial life forms are able to exist in spore form through that boiling, which is most of them. While they may not be active in that form, they can survive. And then once they get back into the correct temperature and environment, they'll awaken and they'll start to multiply. And now they're competing. So you have the biology that was on the rice that got there from the field that it was cultivated in, which is most likely not in your native environment. It's the biology from the field that it was grown in, or whatever conditions, wherever. So you're not introducing a native or indigenous microorganisms. You're introducing some microorganism from wherever to now compete with whatever happens to blow in. And I believe that one has an advantage over the other. It's pretty impossible to sterilize organic matter. It pretty much means you have to burn it to ash, right? Because things live within the cell of of living organisms. Bacteria is such an integral part of every living cell that you have to get down to every living cell and burn it out to get it out. So this process of collecting is a competition between whatever is on the rice and whatever's in your environment. And you may or may not get something native to your environment. Chances are it's going to blow into your soil anyway. So I personally don't use indigenous microorganisms or collect indigenous microorganisms as the input. I think that the term itself to explain what you're doing is improper, because you're assuming that you're collecting indigenous microorganisms, and you could be potentially. So it's really just a collection of microbes. It is an inoculant. It's just another form of an inoculant. And I think as a tool, it has its place and time, but I particularly don't subscribe to the concept.
So my view of IMO a little bit, all that stuff what Luna said, all completely accurate. It's, you know, these, if you look at the endophytic microbes that are inside of rice, it's actually a real issue as far as food safety. Like you can't leave rice out for a certain period of time because the endophytic microbes will start blooming and they're food pathogens. You let rice rot in your fridge and it looks like it's mucor. It looks exactly like an IMO collection. So mucor is, and all these other different potential collections that you grow on rice, they may or may not have direct value as an inoculant, but from understanding that microbes use signaling chemicals, microbes, it's called quorum sensing. And a lot of creatures do it, bees do it. You know, all of a sudden the whole nature of a beehive will change off of one little signaling molecule. And they've found that in microbial systems, specifically someone was talking about carrots. You know, you have a bag of carrots in your fridge, and all of a sudden they're fine, they're fine, they're fine. Then one day they go soggy, they just turn slimy. That's a certain species of microbe that's completely covering all the carrots all the time, but they hit a certain point and they hit quorum sensing, and they just boom, and all of a sudden they all change. So there's all these signaling molecules and everything, and I feel like why people see a lot of the benefits of IMO and these other things is you're taking microbes, no matter what they are, they're decomposing microbes, they're creating all these signaling molecules and hormones and peptides and all these different things. And now you're introducing it to a soil where it feeds everything else. You know, a lot of these signaling hormones and chemicals are very similar throughout microbial species. So you're just kind of flushing it with all this stuff, all this food that makes them go, hey, I'm awake. And it wakes a lot of other things up. It's been shown that certain mycorrhizae species don't flourish unless they have a certain bacterial population associated with them. And they're finding all these correlations where you need one of these things in order for something else to happen. You know, it's really a crazy, crazy complex system that's been going on for eons.
It's certainly an interesting tangent, IMO, and certainly people have used it with great success, and it's something I think we should continue to play with and maybe do some side-by-side studies on.
But if you want inoculation, I feel like grabbing some forest soil or just even some soil from outside, like that's an inoculant. You're bringing in all these different microbes. You know they're there. You know, as it stands, we can only cultivate about 1% of all soil microbes, and that's amongst a whole bunch of different types of agars and food sources. So I don't see how rice, a very simple carbohydrate, can collect a highly diverse microbial species collection. You know, it just doesn't seem logical to me. So you go take a handful of forest soil and you mix it in a 5-gallon bucket and water it in. Some things are going to be introduced. A lot of things are just going to die off or just go dormant until the proper environment exists, but you are inoculating, you know?
Well, the one thing that we keep coming back to is kind of building that home, right? So like if you build, whether you bring them in or how you bring them in, the way to keep them there and the way to flourish them is by building this home. So, and the other key word I keep hearing you say is amino acids, Tim. So what is the real world application here? What is our end game? Why are they so important? What are we trying to do with them?
Amino acids are complex nitrogen compounds. They full of carbon and oxygen and all sorts of good stuff. And they are signaling chemicals. They have all sorts of different physiological functions. They're parts of enzymes. They're the basic building blocks of everything that we want. So really what plants point of the whole point of plants is to create, oxygen, but also to create protein. Like that's what they want to do. They want photosynthesis is designed to create sugars and create proteins. So the scale goes from amino acids, then they create peptides, which are multiple amino acids put together. And then when you get multiple peptides put together, you start creating complete proteins. And that's where I was just reading a whole bunch of studies earlier. They're showing increased root growth rates, increased sprout rate, shoot rates, higher medicinal compound levels in aromatic plants. All from just using amino acids instead of raw forms of N. If you also look at every flavonoid and terpene system is based off of a different amino acid. So the way I look at it is, amino acids are microbe foods, they're plant foods, they're the basis of all these different complex compounds. So they, to me, are the ideal input that you could use for cannabis. Anybody I've ever suggested to do anything to try to improve the quality of a cannabis is switched from a raw N form from nitrate or ammonia to amino acids to soy protein hydrolysate, and the quality of their cannabis has gone up every time. It's really incredible.
Luna, how do you go about accomplishing this and keeping your amino acids where you want them to be and incorporated in your outdoor grow?
So like Tim had mentioned, amino acids are a high source of nitrogen. So first of all, in a living soil system, we have to have our nutrient proportions balanced. Each nutrient has a relationship to the one next to it, and I'm not going to get into the specific proportions, but it's important to keep those in mind. So I always get my soil tested to see what my range of nitrogen is. To see how much I can add. And there are certain programs and resources you can use to do those calculations for you. I like to use soy protein, soy-based amino acid sources from like soybeans, right? I also like to use aloe vera. It has a very diverse profile of amino acids. It's only missing one out of the entire profile. Each one of these amino acids has a specific function. Some of them are better at building phytohormones and phytocompounds that signal metabolic pathways, that signal plant defenses, that signal flavonoid pathways. And all of these are, create secondary metabolites inside the plant that express like aroma and just regular functions like budding, elongation of the cells, chlorophyll density. They affect all these things. They create in combination with other organic acids and with biology, they're able to ferment and create these compounds that signal other processes in the plant. So I like to introduce amino acids in the most diverse way possible. So aloe vera is one of my favorite ones. And I'll just take it and I'll blend it and strain it and add it. Aloe vera is also rich in like salicylic acid, which promotes plant defenses, has a lot of B vitamins, has a lot of minerals. It's really great. Hormones and stuff.
Hormones.
Yeah,
But
hormones.
you'll specifically do this based on a soil test.
I'm sorry, you said I only do it based on a soil test?
That's what I'm asking.
So if I have the room to add a bunch of amino acids without pushing my nitrogen levels too high, I'll do it, which is pretty much all the time. The soil mix that I'm using, that Tim and I are working with, is really low in nitrogen. This keeps the temperatures down. It also allows us to integrate a lot of amino acids pretty much whenever we want to. There is a point where it's no longer beneficial and you want to pull back on it. But if you've added enough to get to that point, you have plenty of amino acids in your soil.
So something I heard that was really interesting to me was you're talking about specific amino acids that will affect specific flavonoids or terpenes. Does that mean that if I have a specific desired result in mind, I could add specific amino acids to get to that result?
So it's in relation to the genetic potential of the plant, right? If the genetics are not there to create a specific expression, it's not going to do it. So what it's doing is it's opening up pathways that then allow the genetic potential to be expressed. Um, and this is, you know, our our terroir, right? Our specific topographical expression in the plant, that makes your cannabis unique and set aside from the next one is how it expresses itself. And a lot of the times, if you don't have the entire profile of compounds, amino acids, and all of their conjugates, which are the compounds created from amino acid, the amino acid conjugates, um, you don't get the full expression. So there's the combination of amino acids, the integration with the biology, and that's all microbes, including fungi, protozoa, and how they interact with each other, as well as the sugar sources that you have. It's a full picture that creates the full genetic expression of your plant. Um, so it's not as simple as I'm after this terpene, I'm going to go get this amino acid and I'm going to create this terpene, or I'm after this terpene and I'm after, you know, so I go get this one particular biological source and it creates this one terpene. It's not as cut and dry as that. It's as much as it's more like unlocking its full genetic expression and potential through the integration of a complete system within the soil.
Yeah. So plants always are making amino acids. That's part of their function in life. That's why they uptake nitrogen. And by feeding amino acids, we take away the plant's need as much to do the conversion. So that 10 parts of energy for every 1 part of nitrogen that it tries to convert, it doesn't need to do that any longer. So it also has 10 extra parts of energy and has all these amino acids flooding in. So now it can fully express all these different pathways. You know, that's and then so Luna and I look at these as a nitrogen source because me personally, I'm not confident that I have a proper nitrogen cycle going on in my plant, but in nature, nobody feeds plant nitrogen. We have 80% of the air we breathe with nitrogen. There's something like 1 million pounds of nitrogen over every square foot that is on Earth. And there is no reason why we should actually have to add nitrogen to plants. Now that we're learning about endophytic microbes and the work of Dr. James White and his team, they've actually done a bunch of research on trichomes on cannabis, and they found a nitrogen-fixing microbe inside of the trichome. And one of the things that they theorize is that trichomes are perfectly shaped for air exchange. They have this perfect design where they exchange air very well. And then they have this nitrogen, they should have this nitrogen fixing microbe inside of them. Nitrogen fixing microbes are based off the enzyme nitrogenase. The nitrogenase enzyme gets broken down instantly when it's exposed to oxygen. Cannabinoids are oxygen absorbers. So they theorize that about 40% of a plant's nitrogen capacity come directly through nitrogen fixing microbes inside of trichomes. So how do you get there, though, is from my understanding of agronomists, is you need to earn the right to not buy nitrogen. And the way you do that is you provide a great home and you provide a lot of photosynthesis. You know, all these things that Luna and I are talking about, you know, acids and, you know, peptides and photohormones and all these are all photosynthetic photosynthates. They are the compounds that a plant produces, and a healthy plant will produce 50% more than they need and just send it directly to the soil to feed the roots. But if your plant isn't producing the excess and it's not feeding the microbes in the soil properly, then you don't get the right not to use nitrogen. So the amino acid thing is like, plants are full of amino acids all the time. It's just as a food source, it's best to use amino acids versus raw forms, you know, but they're always floating around. They're always doing their thing.
So that's the living soil part.
Yeah. Yeah, that's it. I mean, it's a cycle, you know. It's the plants, the microbes release nutrients for the plant that creates photosynthesis. It just feeds more microbes to create that circle, just going over and over again.
So circling back a little bit to something you mentioned earlier, Luna, which caught my attention, was this idea of inoculants or microorganisms that maybe we don't want. And it's interesting because when you talk about things like fungus and bacteria, in most of your lives, those would be considered things you don't want, or at least how we talk about them. You know, so we always assume when we're talking kind of in this living soil, these are things that we do want. But can you talk to a little bit about that balance we have and how we kind of are constantly maintaining the good and the bad within our soil?
Yeah, so the bad, quote unquote, is a very it's a very subjective term, right? Um, the bad is necessarily it's kind of difficult to explain. So like it's an imbalance. The bad is the imbalance. It's when one particular microbe is going to be dominating your environment. Um, Typically, this is like in an anaerobic setting. If you have an anaerobic setting mixed with an aerobic setting, you can create plant pathogens like fusarium um, uh, or pythium. These different things that are not going to, that are going to break down the organic matter in the ways that we don't want it to. These pathogens, they're microbes. They are the result of organic matter not being composted or broken down effectively by like the microbes that we do want. They move in nature when these processes aren't happening fast enough because they're very voracious. So if your systems aren't functioning properly to break down organic matter in an effective way, these pathogens, these undesired microbes move in and start breaking things down for you. And then you find that they infect your plant, they interrupt the processes that you want, and they shut them down. There are instances where, when introduced with specific endophytes, specific microbes that can then go inside of these pathogens and change, they actually change the function of the pathogens and can become beneficial, which is kind of like, what does that mean? How do we integrate that? And that's kind of a tricky question because the relationships are so broad, and there's so much research to be done on the specific pathogens and the specific endophytes that interact with them to change their functions. So
Luna,
it's the
can we stop real quick? I think Greg had a question.
sure.
Hi guys, I'm just wondering what's the best way to keep that organic breakdown up and running? Is worms the best option for that?
So it's just about having a proper inoculant, having proper bacterial sources and forms. I like to use insect-based inoculants. I like to use earthworm castings, which will then, they're full of earth or egg worm eggs. They're full of worm eggs, so you'll have worms that start to hatch and grow within about 2, 3, 4 weeks of introducing your earthworm castings. I also like to use insect frass. I do some aerobic fermentations, making compost teas that I'm a fan of. I know Tim doesn't do that very often. I like top dressing earthworm castings and insect frass. I also like introducing bukashi, which is a grain carbohydrate that's been inoculated with lactobacillus. Lactobacillus has a lot of antipathogenic functions. It's one of the most common microbes out there. It has tons of different functions. It secretes proteolytic enzymes that break down proteins into amino acid form. So it has a really nice function. It's what's called an opportunistic organism. So it competitively excludes these pathogens. And that's what we're after, is competitive exclusion, which is a microbe competing for the same food source as something that you don't want. So if we introduce enough beneficial biology, we are competitively excluding the biology that we don't want, because they compete for the same resources. So if you occupy a space with all the good, it kind of keeps the bad from having an opportunity to come in. So it's about maintaining microbial density in your soil. You can achieve this through microscopy. But most of the time, if you build your soil properly with the correct inoculants and with quality inoculants that are biologically active, you don't need to worry about using a microscope. That's kind of next level stuff that you can get into, where people like to nerd out. I like to do that a lot. I'm really into it. I find it to be fun. So it's just about providing the inoculant and feeding the inoculant a food source to keep your beneficial biology functioning, as well as maintaining an aerobic environment in your soil. You know, you don't want to overwater it. You don't want to let it dry out and underwater. You want to keep, it's about balancing the function of biology, of bacteria, of protozoa and fungi in your soil through correct moisture, correct feeding, inoculants, and the correct profile of nutrients. So testing, inoculating, feeding your biology. Yeah.
Air exchange, gas exchange, gas exchange
Yeah.
is huge. You know, you can have anaerobic, you can have aerobic, and both of them can have highly beneficial properties to them. But it's when nobody can really breathe and nobody can really do anything. You know, you can't let the aerobes benefit, and the anaerobes aren't. There's, you know, you want to. Gas exchange is the number one. And then one comment on getting into like beneficial and harmful microbes, like fusarium. Fusarium is incredibly common. It's everywhere, all the time. Can't get rid of it. It's a strapophobe. It's a natural breakdown thing in most soils. But as soon as it finds a healthy plant, it switches over to being a pathogen. And they're finding, some research realized that like some of these endophytes that are in plants, even like boritis or some of these other things, they could all of a sudden just trigger and turn on. They could already be endemic inside the plant, and they're there. There's other beneficial microbes that aren't allowing them to do what they do, or their genes aren't expressed to be pathogenic at that time. So
Do you think it's environmental that's triggering it?
Environmental largely, yeah, too, because you have all these other microbes like the rhino. Bad at pronouncing these, but the microlife product, like that microbe is shown to turn on and off genes that will stop microbes becoming pathogenic. And the lactobacilli specifically, I was just looking them up, they just completely reclassify the entire lactobacilli family because they realized how huge the genome was. And the genome can just change and turn on and off. And they could do all these crazy things and survive in all these crazy environments. They're actually recently been combining microbes. They thought one microbe was over here, one microbe was over here. They look at the genes, you realize it's the same microbe. They're just expressing different genes at different times based on their environment, on the surrounding microbial community. It's heavy. There's so many things at play. Like for me, like air exchange and providing a food source, and you keep the right people happy. You keep the right ones happy, and everyone else just stays at bay.
That's brilliant. So I think this is a great point to see if there's any more questions and kind of wrap up with some real world advice there. You could just
Johnny,
talk.
go ahead. Yeah.
Hey guys, great talk. Love everything that I'm hearing here, and I can't believe other people have these thoughts.
Nice.
So yeah, calling into question IMO is like what I've been trying to do. So realistically, when we water, we force a compaction over time, and most of us who are trying to do a living medium, we're reusing these mediums for long periods of time. What about the level of compaction and the effect on biology, and maybe some tactics to avoid compaction in your containers or your beds?
So if you're in a traditional soil, like a clay-based soil, calcium, lots and lots of calcium. Open up that, flocculate that soil, you open it up. If you have a heavier sandy soil, then you don't really need to worry about it as much. But clay soil is calcium all the way. Calcium will flocculate the clay plates of the clay and spread them out, which allow more gas exchange, which will feed microbes and allows everybody to be happier. Indoors, I've been running same soil for 11 years or so now. And I find that it turns into all organic matter. And every once in a while, I kind of have to fluff it no matter what I do. I've been experimenting both ways. It's hard. You have to have a really strong microbial community and lots of macroarthropods and earthworms and everything else in order to till that soil and to create that aggregate in the heavy organic matter, because it does inevitably just compact down, especially peat-based soils and stuff. Or you got to flip them once a year or so and just add some more aeration in there. I haven't found a real good thing other than really, really, really strong biology to keep it fluffy.
Thanks, great
Yeah,
answer.
also, earthworms are going to play a big role into promoting an aerobic environment. The degradation of root systems also opens up, air exchange. So you have, your roots. When in a living soil system, you grow your plant, you cut your root ball out, right? And you yank it, and I'll just plant, either right next to it or in the same hole. And as the roots grow, they kind of fill in the same channels as the roots that start to break down, and then they can immediately plug into those mycorrhizal networks and stuff. This also functions with cover crops and stuff too. But you have to take the cover crop down, let it decompose, and it opens up those channels also. Like Tim said, over time, it's going to compact. You might just have to fluff it a bit. Sometimes people like to take pitchforks and just like stab at the soil, just stab it real good if you don't want to fully till it, because I know in no-till living soil systems our mycorrhizal and our mycelial networks are super important. The myco-transportation of these hyphal networks are a big integral part of how these systems function and how nutrients are transported from one particular spot to another, and maximizes the root zone reach through, extending the surface area of the root zone through these mycorrhizal systems. So we try not to, till it as much as possible. So like stabbing it, keeping, like Tim said, keeping your full profile of life in the soil, which is your insects, your microarthropods, the soil food web. So that's mostly it. Just encouraging the full spectrum of things that you need in nature. We have grazers. We have like the animals that come along and they eat the companion plants, the cover crops, and then they kick and then they poop, and they dig for the worms and they dig for the bugs and they eat it. And we don't really have that in an indoor setting, and we try to keep things like chickens and stuff out of our gardens. So we kind of have to fill in that role, because we don't really want animals around our cannabis plants being dirty and stuff. So sometimes you just kind of have to do it.
Good reminder. Lucas had another question.
I got a question about, do you have any recommendations about literature where you like to get some basic understanding? Like I got some books from Jeff Lowenfels. I mean, I don't know. Is this a good start where to go and stuff?
The first I haven't read the fourth one, but the first three are gold. And I'm sure the fourth one is as well. But that's like base, hands-on, the most approachable. They're gold. That's the best place to start.
Good. And do you have any podcast recommendations or whatever? I'm listening to Builder Soil. I don't know, like what do you think?
Builder Soil is good. Kiss Organics, Tad's Science and Cultivation podcast. His is
I know this one.
Hands down my favorite.
Good.
And
John
then
Kemp.
John Kemp. Anything from, so if you really want to dig deep into soil biology, and this is more like broad agriculture, this isn't cannabis stuff.
Yeah,
John Kemp. Anything from John Kemp and Advancing Eco Agriculture, which he has a lot. a lot of information out there.
KAMPF.
KEMPF.
Kemp. Okay.
Kemp,
good.
John Kemp.
Thank you.
And then
I have Chris Trump.
Chris Trump's good.
KNF. Or...
So he's the KNF stuff. Chris is very knowledgeable, and he definitely knows how to work with environmental systems.
but
he's really specific to KNF though, which is great.
Okay,
and then one thing that's a really great resource is the Bionutrient Food Association. It's run by Dan Kitteridge, and they have had a conference for 11 years or so, and they have the past 8 years or so online. And you could probably spend the next 2 years watching videos on there and never watch them all. It's incredible the resource that is. All different speakers, talk about plant health, human health, environment, water, you name it, they talk about it. And it's all based on, nutrients and food and helping human health stuff and helping the environment. But that's
good. Thank you so much, guys. Really loved it.
No problem.
Amazing stuff.
Yeah, no problem.
Luna and Tim, I just want to say thank you guys so much for doing this for us, and I really appreciate it. You guys are brilliant, and I appreciate everything you guys do, and especially the bud you guys both grow.
Thanks. If I could just shamelessly plug myself real quick. So I do teach a lot of these concepts. I share a lot of white papers, research that I come across, as well as PDFs with a lot of this knowledge in my Patreon channel. And you can join the Discord server where I just dump tons of information on a lot of these concepts. So if you wanted to connect with me, I'm more than happy to educate people.
Can you tell us how do we find you? What's your name on Patreon and what's your Discord?
So I go by Luna All Day is my Instagram channel. So it's LUNALLDAY. And through there on my bio, there's a link that takes you to all of my different social media, including Patreon.
Perfect.
And you can connect to it that way. You can always reach out to me directly. I'm fighting COVID right now, so I have tons of free time on my hands. If you want to reach out to me, I'm more than happy to help you and send you resources.